Welcome to The Explainer. Today, we're diving into a genuinely groundbreaking study by Dr. Mohan Saxena and Sugindha Raj. They're tackling this massive, yet honestly totally overlooked global health and economic threat. We are talking about a literal monumental shift in veterinary medicine here, and it's one that actually has some incredible implications for human health too.
To kick things off, we really have to ask a massive question. How do you actually cure a disease that historically turns animals into lifelong, highly contagious carriers? I mean, for decades, veterinary science has struggled with these bacterial infections that are just stubbornly, fundamentally incurable. They totally refuse to be cleared from the host, no matter what kind of treatments we throw at them. Okay, let's start with the actual victim of this disease, the cow. If an animal contracts bovine brucellosis, which is caused by the bacteria Brucella abortus, it basically faces a lifetime of suffering. We're talking about devastating abortions in the last trimester, severe infertility, and these really painful joint swellings called hygromas. It's just a massive blow to animal welfare, not to mention agricultural economies worldwide. But the real danger here goes way beyond the pasture. Brucellosis is a zoonotic disease, meaning it can jump right from animals to humans. Because the bacteria shed so heavily in an infected cow's milk, anyone consuming unpasteurized milk or even just coming into direct contact with it is completely at risk. So this isn't just an agricultural headache, it's a really critical public health and food safety crisis that demands an actual cure, not just management. Right now, we kind of try to control this with two live attenuated vaccines, S19 and RB51. But they have some glaring flaws. S19 only induces antibodies, which, as we'll see, are largely useless here because Brucella literally hides inside the host's cells. Worse than that, S19 can actually cause the very abortions it's meant to prevent. Now, RB51 is a bit better because it triggers cellular immunity, but there's a terrifying catch. It's highly resistant to the antibiotic rifampicin. So if a human accidentally gets infected by the vaccine strain, treating them becomes an absolute medical nightmare. So to build a better weapon, the researchers went straight to the lab and grew fresh cultures of both these flawed vaccine strains, S19 and RB51. Here is the actual bacterial growth from their lab work. But the goal wasn't to use these live bacteria. Instead, they wanted to harvest them to create something entirely new and vastly safer. Then they introduced a specialized Brucellophage to those cultures. Now, if you don't know, a bacteriophage is basically a virus that specifically targets and destroys bacteria. Those clear dots or plaques are where the phage has literally popped the Brucella bacteria like balloons. It destroys the live threat, and it leaves behind this highly recognizable debris field. In the science world, we call this destroyed bacterial debris a phage lysate. And honestly, here is where the absolute genius of this approach lies. Think of this phage lysate as the ultimate training dummy for the cow's immune system. Unlike killing bacteria with heat or chemicals, which kind of melts and warps their shape, the phage pops them open, but leaves all their original recognizable parts totally intact. So it triggers a massive immune alarm without any danger of a live infection or the awful side effects of those traditional vaccines. So, how does this actually hold up in the real world? Well, the team took this out of the lab and ran a 90-day trial with 21 naturally infected cows. They divided them into groups, giving them just a single 2-milliliter injection right under the skin. They used either the S19 lysate, the RB51 lysate, or a cocktail of both. They also left a control group untreated to compare, and then they meticulously monitored all of them for 90 days. The immune power unleashed by this single dose? Absolutely incredible. Notice the steady, really robust climb of anti-Brucella antibodies over the 90 days. That top line represents the cows treated with the cocktail lysate containing both strains. Their humoral immune system just kicked into absolute overdrive to fight off the infection.
But remember what we talked about earlier? Antibodies just aren't enough for an intracellular pathogen like Brucella because it hides out inside the cells. So the researchers also tracked total leukocytes and neutrophils, basically the foot soldiers of the immune system. The data here shows significant increases in these cell counts for the treated cows, which tells us that a really strong, broad cellular immune response was triggered right alongside those antibodies. And that brings us to exactly what we want to see. This ELISpot assay from a cocktail-treated cow is literally lighting up with red spots. Each one of those spots represents a lymphocyte-producing interferon gamma, which is essentially an immune system distress signal and weapon all rolled into one. This is just stunning visual proof that the cattle's T cells are activated and actively hunting down the Brucella hiding inside their cells. Okay, so the immune system is clearly fighting back. But that raises a massive question. How do we definitively prove these cows are actually cured? I mean, how do we know the live bacteria are truly gone without subjecting the poor animals to highly invasive tissue biopsies? To figure this out, the researchers used a really innovative, non-invasive biomarker. They didn't just look for antibodies, they looked for a very specific 223 base pair genetic footprint of live Brucella RNA directly in the cow's blood plasma. Because RNA degrades so quickly, finding it means live bacteria are actively shedding it right at that moment. Let's look at the evidence here. Early in the trial, right at day 0, look at those bright, glowing bands across almost all the lanes on this gel. That is that exact genetic footprint we just talked about. It visually confirms, right out of the gate, that the cattle in the trial are heavily infected with live Brucella organisms. But man, watch what happens by day 75. Look closely at the lanes marked by arrows. Those glowing bands are fading away, becoming nearly completely invisible in the cows treated with the cocktail lysate. Their bodies are clearing the live bacteria. Meanwhile, on the right side, the untreated control cows still have these bright, thick bands. They remain heavily infected. By day 90, the results were just staggering. The researchers confirmed that the Brucella RNA biomarker was completely undetectable in the treated animals. And they even went a step further. Actual blood and tissue cultures from vaginal and uterine secretions showed absolutely zero live bacterial growth. The pathogen was completely eliminated. So, we are left with a pretty provocative question to consider. Based on this incredibly robust, heavily documented data, is this finally the end of the global brucellosis threat? Have we actually found a way to clear a disease that was previously thought to be a lifelong sentence? Well, the evidence for sure points in that direction. This explainer really highlights a monumental medical breakthrough. We're talking about a safe, one-dose phage lysate cocktail that acts as a true therapeutic cure. It not only clears an incurable infection, protecting animal welfare, but it safely guards our human food supply from a highly dangerous zoonotic pathogen. It's just a stunning victory for targeted, non-invasive immunotherapy, and definitely something to keep your eye on.